One of the four biomolecules are the proteins, and proteins are polymers made up of repeating units of amino acids. So given that amino acids are the building blocks, let's start with that. This is the general structure of the amino acids relevant to biochemistry, and as the words amino and acid imply, they have functional groups relevant to these. The amino group here, which is this one, and the carboxyl group here, which as we know, things with these are called carboxylic acids. Now, there are properties that are more or less generalizable for majority of them, and we will discuss them first. Before we go to the individual amino acids, first, amino acids are actually known to be amphoteric, and amphoteric means that it is both acidic and basic, or it has the potential to be either acidic or basic. And I think that requires us to recall what the definitions of a base and an acid are. For this case, we will use the Bronsted-Lowry definition, which hopefully you would remember has something to do with the release or the uh, accepting of protons. So for example, we usually call things with carboxyl groups as carboxylic acids, and that is because, long story short, they are proton donors. And if they donate protons, the COOH will get rid of this H as a proton H+, and we will be left with the functional group COO minus. And since it also has the amino group, as we recall in organic chemistry, amino groups are actually basic such that they can accept a proton, an H+, giving us something like NH3 plus at the end. Next, amino acids are chiral, and this is something we've already seen in carbohydrates. We can look here at this carbon, see that it has four substituents, and assuming that the R here, the variable group here, let's name this variable, meaning it changes depending on the amino acid. Assuming that the variable group here is not the same as any of the three other substituents here on top, then those all four are different, and that would make the central carbon chiral. Remember, when we have chirality, therefore, it will give rise to isomers. Do you remember in carbohydrates when we discussed the D and the L isomers? So we have the D isomers and the L isomers. Now, if you do, maybe it would help you if I say that this one is a structure of an L amino acid. Given that, can you tell me what is the basis for giving the D or the L designation for this? If you remember before, when we give the letter L in carbohydrates, it means something you're looking at is on the left. If it's D, something you're looking at is on the right. Since the only thing here in the structure that's written at the left part is the amino group, that means our basis for the D or the L letters is the position of the amino group based on where it's drawn. Okay? And it so happens that the L isomers are the ones that are common in eukaryotes, and this means that for us humans, most of our proteins are made up of L amino acids. The isomers are not impossible to find, but they are less likely to be found in us. We can find this actually more of existent in prokaryotes and some bacterial cells, but we will not deal with that too much for now. 20 of the amino acids that we will talk about are proteinogenic. When you say proteinogenic, these are the only amino acids, 20 of them, that can give rise to proteins. And that is actually based on something that we call the genetic code, which is something we will discuss further when we get to our DNA and RNA. But the point is, you have to realize that the word amino acid is actually a very generic term. Because if you think about it, anything with an NH2 and a COOH are called amino acids, but that doesn't mean that all of the compounds in the universe with that basic requirement is something that we can use for proteins. I can just assume we have hundreds or even thousands of amino acids in the entire universe, but we're not going to memorize 1,000. That's crazy. It so happens that in our DNA, we have only 20. Yeah, 20 is a lot, but let's just assume it as few compared to, I don't know, 1,000. And the point is, now what we're going to do is to discuss these 20 individual amino acids. And what we're going to do is to classify them. Let me write that here. Classify them first, because there are other classifications later, by their R group, and specifically by the polarity of the R group. So it is a given that at this point we already know if something is polar or nonpolar. Now, I present to you first the 9 nonpolar amino acids, and you can see that this portion right here is already written in white every single time because it's more or less constant. You're always going to see this NH2 and the COOH in every single amino acid because that's where we get the name from anyway, right? And the only thing that's colored here is obviously the R group, which is intended for you to see the difference among these. Let's start with the one here because it has the smallest R group. I mean, what could be smaller than a single hydrogen atom, right? And the amino acid that contains the R group of H is called glycine. Now, take note that for our amino acids, we actually have 3-letter and 1-letter abbreviations. So for glycine, its 3-letter abbreviation would be GLY, and 1 letter would be G. Okay? And be careful because later on you will have another amino acid that is almost same as GLY. Now, going to the structure of glycine, you have to recognize that we already had a hydrogen in the constant portion here above. And if I have my R group as a hydrogen, that means that my carbon in the middle has 2 similar substituents. And you know what that means? This one is not chiral, or in other words, a chiral. And in fact, among the 20 proteinogenic amino acids, this is the only one that is a chiral. So given it's a chiral, it's impossible for us to call something like L-glycine or D-glycine because you can only reserve these letters for chiral compounds, which glycine is not. Next, if we have a methyl group as the R group, the name of this amino acid is alanine. 3 letters ALA, 1 letter A. Simple as that. Then here, this is intentional. As you see, another methyl group is present. So this would have been alanine. But the next amino acid, just for you to see the relationship with alanine, is almost like alanine except one of those hydrogens is removed and replaced with a benzene group. And remember, when we name organic compounds that have a benzene substituent, the name is phenyl, right? Phenyl. So all we have to do is to copy the name alanine and add the prefix phenyl. And we have the name phenylalanine. 3-letter abbreviation PHA. Now, there are some cases like for phenylalanine that the first letter of the actual name is not the 1-letter abbreviation for reasons you will see later, because for phenylalanine instead of letter P we will have the letter F, which I hope makes sense because when you pronounce phenyl in the first place, this sounds more or less like an F. Okay? Next, we move here and we see more carbons, at least compared to alanine. This has 3, 1, 2, 3. If you are a little polished with naming rules, you can call this the isopropyl group. So if you see the isopropyl group, which kind of, coincidentally forms a letter V shape as the R group, this is actually the amino acid valine. Sorry, no, I'm correct, valine. I said it's coincidentally because, well, it's shaped like a V upside down and it's valine. VAL or letter V. Okay. Then beside it is almost the same. You see, it's like valine with an extra carbon here. And the name of this one is isoleucine. 1 letter is ILE, and sorry, 3 letters is ILE, 1 letter is I. And because ISO is already an official prefix in the IUPAC naming rules, we don't use the prefix ISO for isoleucine also. It would be confusing. So instead of these 2 letters, we move forward to the next 2, giving us ILE. Actually, of course, there are other ways to explain why it's like ILE, but that's, I think, one of the more logical ways to put it. This one is called leucine, and 3 letters is LEU, 1 letter is L. Okay. Now, I actually have a lot of comments for these, but let's get back to that after I finish these three below. Here at the bottom, you have first this one. I have a CH2 CH2 an ethyl group here, followed by a sulfur atom. So this is quite unique because we don't normally see sulfur, right? So this is one of the very rare cases. And then a methyl group at the end. Now, if you're gonna name this SCH3 group, you're supposed to name it from the methyl going inside, right? If you do that, the name of this thing would be, or at least this SCH3 portion would be methyl thio, methyl, and then sulfur. And actually, I don't really need to say this, but I prefer to say this because it will give us more idea on why its name is like this. Methionine. Three letters, M-E-T, and one letter is M. Next, we go here, and we have a methyl group with a ring system right here. If you already are familiar with the common heterocycles, you may be familiar with this. Some of you may even remember its name. But just in case you don't, the name of this ring system is indole. And this indole containing amino acid is called tryptophan. Tryptophan is one of the most annoying in terms of abbreviations because its three-letter abbreviation is TRP, not TRY. So this is already not kind of directly, lifted from the name. And what's more annoying is its one-letter abbreviation, W. And really, there's a lot of crazy stuff people try to do to remember that it's letter W. They're like, oh, let's try to force the issue. Let's like call it tryptophan, and people even do more crazy stuff about this. But whatever, really. Just remember that it's literally W. That's what's designated for tryptophan. Next, for the last of the nonpolar amino acids, we have here the R group, CH2, which is connected to another CH2, which is connected to another CH2. And this one is weird because it now goes back to the nitrogen of the constant portion here. And meaning one of these hydrogens would have to be removed, of course. And this is the first and the last time you're going to see this. The name of this amino acid is proline. One letter PRO, sorry, three letters PRO, one letter P. Okay. Now it's time for me to add comments to the amino acids here. First, phenylalanine contains a benzene ring, right? And if you actually notice the indole ring system, one of the rings in indole is also benzene. That's why it's acceptable for you to call tryptophan and phenylalanine as aromatic amino acids. And there's really an importance to that. It's not like just novelty or like mental exercise, because sooner or later there will be a role that aromatic amino acids are kind of special with, or at least it's another convenient way for us to remember grouping phenylalanine and tryptophan together. Speaking of grouping together, there's actually a group wherein valine, isoleucine, and leucine are involved. Notice that their skeletal structures here are branched, right? Because if I start from this carbon and I go here, this would be a branch right there. If I go from this carbon going to the right, there's a branch here. If I go to this from this carbon going down or up, regardless of where I go, there will always be one branch. And therefore, many references actually call these three as the branched chain amino acids. In a way, they're actually metabolized with a similar enzyme, but let's not get too ahead of ourselves. Just call these three VIL as the branched chain amino acids. Now, also, you would notice that isoleucine and leucine are obviously related, just given the name here. And it makes sense because if you count the carbon of their R groups, they're both four carbons. So they are butyl groups, right? Four carbons. For isoleucine, its butyl is actually a sec-butyl, if you know your naming. It's a sec-butyl attached to this carbon. But for this one, given it has a forked shape at the end, it's actually an isobutyl group. Okay, so it's not really something that is taught at the beginner level unless you're in some hardcore chemistry major course. But I thought it would be nice to mention it as well. One last thing about proline. I did mention something special about it, right? The fact that proline is the only amino acid that closes back to its amino group. And that's weird because given that we have the amino acids here, except proline, all of their nitrogens, except for proline, of course, all of the nitrogens here are primary amines, right? Because they are only attached to one carbon, and that's the chiral carbon here. So this is the only secondary amino group existent among the 20 proteinogenic amino acids. Sometimes books would prefer calling proline an amino acid because of this special phenomenon or special structural feature. I think that's all for the non-polar amino acids. And just to give us one final look, notice that they all have almost only carbons. I mean, there are only a few exceptions to that. Yeah, we have one nitrogen from tryptophan, we have one sulfur for methionine, but I think you could forgive them given that the remaining substituents are all carbons. And isn't it when an R group has carbons only? Carbon, 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 everything here is carbon, carbon, benzene is full of carbons. It's non-polar. So hopefully it makes sense. Next, we have 11 to go. These are just 9, right? Let's go. So here we go to the next amino acids, and as you notice, I kind of color-coded them just to set them apart. The ones here at the left are called the polar amino acids, and I think this would make sense because if you look at their R groups, there are already electronegative atoms like oxygen, not just oxygen, a hydroxyl group, which is highly polar, a thiol group, which is also highly polar. Here we have OH again, and we have CONH2, so amide groups, which of course contain all those electronegative atoms and again are, once again, let me repeat, polar. Not something you see in the nonpolar amino acids a while ago. The one with a methyl group, then an OH at the end, is called serine. 3 letters SER, 1 letter S. Then actually there's something almost the same as serine. We have a carbon just like serine, an OH just like serine, but with an extra methyl group. This one is threonine. And for me this is very convenient because they're beside each other in the alphabet. After letter S you have letter T, so I can think of them as like almost the same as well. Then here, the one with the thiol group, so it's literally just like if you have serine, you replace the oxygen with the sulfur. This one is called cysteine. 3 letters, CYS, 1 letter C. Now this one, I want you to pause and then go back to the nonpolar amino acids and notice that this is really almost a lookalike of something a while ago. So actually, if you go back here, it's almost the same as phenylalanine. This is the R group of phenylalanine, but this one has an extra OH. So of course, it's not exactly the same. The name of this amino acid, which is a very different name from phenylalanine, by the way, tyrosine. This is called tyrosine. 3 letters TYR. And of course, since letter T has already been taken by tyrosine, we will give it the letter Y, which is probably the most appropriate letter given that Y is the second letter in the name. Now, since we have here a benzene with an OH, we should not ignore that. Remember, we call this a phenol group or a phenolic group. And in fact, we can say that tyrosine is the only phenolic amino acid among the 20. And also, given the fact that it has benzene, just like phenylalanine, it will not escape our adjective aromatic. So actually, when you think of it, we now have 3 aromatic amino acids total. Tyrosine, this one, phenylalanine, and tryptophan. Then we have our 2 amides below, and in fact they're almost the same. Here, the amide at the left has only 2 carbons. Here, the amide at the right has an R group with 3 carbons, so it's just a single carbon difference. The shorter one is called asparagine. 3 letters ASN, 1 letter N, and then this one is called glutamine. 3 letters GLN, 1 letter Q. So I think given that, amides have the amino group, the nitrogen atom, you can probably think that the third letter would always be N also. because for a reason I will say later. And also their one letter abbreviations are very unique, pretty much like tryptophan. So maybe you could make your own mnemonics for this. But yeah, these are the two amide amino acids. So these are the six polar amino acids. So if we add so far the numbers, we have 9 here in the nonpolar, and then 6 here, we have 15. So we have 5 remaining, and these are at the right, 1, 2, 3, 4, 5. But these are actually further divided into two. The ones color-coded in red are called the acidic amino acids. And I think it's very easy to know why. Given that its R group contains a COOH group as well as the one at the right, then this is the reason why we call this an acidic amino acid. And that's weird, right? I mean, let me mention that word again or term again. Acidic amino acid. I mean, if you try to pronounce or to read this like 5 times, it would kind of be funny because you're repeating the first and the third word. But of course, let me clarify this. Isn't it that when we use the word amino acid, this base word, we're referring to the amino group here of the constant portion and the acid group here of the constant portion. So, this adjective at the start, this first acidic word, does not apply to this one, but of course to the R group, which is the point of calling them acidic amino acids. So that should be clear. And now I want you to look at these two and the amides we have here at the bottom. They're almost like the same, right? CH2CONH2, CH2COOH. It's just like you replace the amino group here with an OH and you're done. And I want you to remember it like that because its name, for example, the name of this one, almost similar to this, is aspartic acid. Aspartic acid. 3 letters ASP, 1 letter D. This one has 3 carbons, just like here, and we just replace the amide with a COOH, and this is our glutamic acid. GLU, 1 letter is letter E. And this one, in terms of the first letter, or in the 1 letter abbreviation, is easier to memorize because, once again, they're beside each other in the alphabet. Letter D for the shorter one, letter E for the longer one. So I think that's one way for you to quickly remember it. Now, remember when I was telling you a while ago that the third letter of the amides would always be letter N? Yeah, that's because if I'm gonna use the ASP as the 3 letter abbreviation or GLU as the 3 letter abbreviation, that would actually be possessed by the acid versions, not the amide. Which if you go back here, have the letter N. Alright, one more thing. If ever aspartic acid ionizes, meaning the COOH would become COO minus, doesn't it make sense that we're gonna have to change the name also? So remember this, in the ionized form, when it has as a negative charge, the name of aspartic acid will be converted to aspartate, and in the ionized form, glutamic acid will be called glutamate. So it's just like me telling you that, sometimes the words aspartic acid and aspartate are interchangeable as well as glutamic acid and glutamate. Okay? Now, for the last 3 amino acids color-coded in blue, it would be sensical to call them the basic amino acids because they have a basic R group with them. Remember, the basic functional group is the amino group, which you're actually gonna see in all 3 of them here, here, and you have a lot of choices here. It doesn't really matter for now at what specific nitrogen we would have the charge later on. But for now, let's just focus on the structures. For the first of the 3 basic amino acids, I have a methyl group and then this 5-membered ring heterocycle here. And of course, this has a name. The name of this one is imidazole. And the imidazole containing amino acid is called histidine. Histidine. 3 letters is HIS, 1 letter is H. Now, let me warn you that this is not the same as the ever popular histamine when you're thinking of allergies, but we are actually very, very close to histamine. Not exactly the same though. Now, let's move to this one. We have 4 carbons here, And then at the end of the structure, at the fourth carbon below, we have the amino group. And the name of this amino acid is lysine. 3 letters, LYS. 1 letter is K. Okay. Of course, the letter L has already been taken by leucine. So, for some, I'll not share my story already, but for some reason, they chose the letter K for lysine. And then finally, we have another long chain here, 3 carbons first, followed by some sort of, nitrogen-rich portion here, where you have basically a carbon at the middle that is attached to 2 nitrogens, a single bond, and then double bonded to another. There's actually a name for this functional group. This is called a guanido group or a guanide portion or guanide group, and the only guanido containing amino acid is called arginine. 3 letters, ARG, 1 letter is R. And again, let me repeat, the reason why they are called basic amino acids is that in their R group, there are basic portions, particularly nitrogen atoms, which we will focus more later in another discussion. But for now, it's at least good for us to see all of the 20 amino acids being named and described. However, there are other ways of classifying them. So other than classifying them based on their R group, we can classify them first on being essential or non-essential. So when you say that an amino acid is essential, it is required in diet, meaning you have to eat food that are rich in these amino acids. And that is because while we need these amino acids to make protein so that we would survive, ironically, our body cannot make those. So, since we cannot make them but we need them, we need to get them somewhere else. And I think that's the reason why we eat in the first place. If we are self-sufficient, we don't need to eat in the first place. So, that's the meaning of essential. For non-essential, then those are things that we don't need to eat because in the first place we can synthesize them. So, what's the point of being desperate for it somewhere else when we have it already? Now, there's an age-old mnemonic for the essential amino acids. Actually, this is more complicated than it looks, but let's not go to the gray areas for now. The essential amino acids have this very generic mnemonic PVT or like private Tim Hall. And a lot of the letters here are self-explanatory because, for example, there's only 1 letter V among the amino acids, only 1 letter I, 1 letter M, right? Valine, isoleucine, methionine. There are 2 letter L's here, but there are only 2 letter L's in the first place, leucine and lysine. But there are some letters here that you just need to be careful a bit. For example, letter P can refer to phenylalanine or proline, and it would help you to remember that the P here is actually phenylalanine. There are a lot of letter A's in the amino acids, in fact, like 4 of them, but the A here stands for arginine. And for the 2 T's here, there are 3 letter T's in the 20 amino acids: threonine, tyrosine, and tryptophan. But the two here are only tryptophan and threonine, meaning there's no tyrosine. You have to be careful because usually this is a trick question, which is an essential amino acid, and then people slip with this tyrosine a lot. Okay. Now, one last way of classifying amino acids, which would make complete sense when we get to metabolism, is being glucogenic or ketogenic. Normally, when we are in the fed state, meaning we regularly eat 3 times a day and whatnot, we don't really need to perform things that I will explain for now because our glucose is sufficient to keep us alive. But in times of extended fasting, like a day or two, our body tries to find other ways to give us energy other than sugar. So, in worst case scenarios, our amino acids are mobilized to become something else. Some of them are converted to glucose, or basically our blood sugar, and some of them are converted to things called ketone bodies. We will just discuss this in the future. And whatever amino acid can be converted to glucose is called glucogenic. Whatever can be made into ketone bodies are ketogenic, which are both things that our body can depend upon when we are starving. Now, we have mnemonics for this. For the exclusively ketogenic amino acids, we have the 2 L's, leucine and lysine. meaning the only thing that you can make out of these two are ketone bodies, not glucose. Now, we have some flexible amino acids, meaning depending on what the situation calls for, they can be made into glucose or ketones, and they have the mnemonic fit: phenylalanine, isoleucine, and the three-letter T's. Take note, I just told you there are only three-letter T's in the 20 amino acids. So far, these are 7 amino acids. So, what's it for exclusively glucogenic? Well, this is the only remaining category, right? So, all the 13 remaining amino acids that are not listed below are under exclusively glucogenic amino acids. And if you think about it, including the both glucogenic and ketogenic, 13 plus 5, almost all of our 18, right? Almost all of the amino acids that we have can be converted to glucose, but only a few of them can be converted to ketones. And to appreciate this better, you have to wait for our discussion on carbohydrate and lipid metabolism. For now, we're done with our common ways of classifying amino acids as you would usually encounter in an undergraduate biochemistry course.